Hello students, welcome to E-Pathashala. I am Dr. M. Vasanthi, Professor and Head, Department of Biotechnology, Kamaraj College of Engineering and Technology, Vidya-mitra, Tamil Nadu. We have been doing a lot of modules 14, 15 and 16 on lipids, and the last part of this module is functions of lipids. Again, a quick recap of our understanding of what lipids are. They are fats and they play many important roles in the body, like providing energy to producing hormones. They help to digest and absorb our food properly. And they are believed to have much more diverse and widespread biological role in the body in terms of their intracellular signaling property and local hormonal regulation. The lipids are synthesized in the body through complex biosynthetic pathways. However, there are also certain lipids, as we know already, they are considered essential and they need to be definitely supplemented in the diet. In proper amounts, lipids are a healthy part of our diet. So, now let us understand the different functions of the different lipids. To begin with, since we have studied the structure of triglycerides first, let us also study the function of triglycerides. A triglyceride, which is often called as a triglycerol, triacyl glycerol, is a fatty acid trimester of glycerol. Important for human health in that they are the components which provide most of the lipids in our diet. It says as a solid state at room temperature, the degree of saturation and the length of their chains attached to the glycerol backbone determine their state at the room temperature. So what are the role of triglycerides? Their main role is they are the storage forms of fatty acids. Let me put it in a quote. It's a very important objective type question, periodically and routinely asked in most of the national level examinations. The storage form of fatty acid is triglycerol. The triglycerides are esters of the trihydric alcohol, glycerol and fatty acids. What is their significance? Triglycerides play a very important role. They can be used as an energy storage form. This food reserve can be called upon during periods of starvation with a high calorie content of the fatty acid, adding to the value of storing fat and providing much needed energy. You must have been familiar with the term beta oxidation of fatty acid and how one molecule of fatty acid, palmitic acid, can generate 129 ATPs. So beta oxidation of fatty acids liberates a huge amount of ATP, and that is the important function of triglycerides. In addition to the storage form and energy reserve, the triglycerides can also provide insulation for animals in the form of body fat, which helps them to survive in colder temperatures. The next important class, phospholipids. They are regarded as the derivatives of the phosphatidic acid, in which the phosphate is esterified with the hydroxyl or a suitable alcohol. We know they are very important components of membranes, micelles, liposomes and emulsions. I am sure in the earlier modules we have time and again reiterated on the importance of phospholipid as components of the lipid bilayer membrane. But now we are going to see its additional properties, the micellar nature, the liposomal structures they form and the emulsions. That is a structure of a liposome showing the aqueous cavity at the center of the fatty acid bilayer. A bilayer of such amphipathic lipids has been regarded as a basic structure in the biological membranes. When a critical concentration of these lipids is present in an aqueous medium, they tend to form micelles. They can exist as a bilayer, but after a particular critical concentration in an aqueous medium, the phospholipids tend to form micelles. The aggregation of bile salts into micelles and liposomes and the formation of mixed micelles with the products of fat digestion are important in facilitating absorption of lipids from the intestine. Let us go a little slow. What is the important function of phospholipid? With the aggregation with bile salts, they can form micelles and liposomes. What are the direct advantages of micelles and liposomes? They are essential for facilitating what? The absorption of lipids from the intestine. So one fat in a particular form called micelle or liposome is required for its own absorption, the lipid from the intestine. Another important lipid, cholesterol, which is found in the cell membrane. It's a sterol, which means it's a combination of steroid and an alcohol. Apart from being a component of the cell membrane, we also know that they are essential for the synthesis of sex hormones, estradiol and testosterone, as well as other steroids such as cortisone and vitamin D. In the cell membrane, the steroid ring structure of cholesterol provides a rigid hydrophobic structure that helps boost the rigidity of the cell membrane. So without cholesterol, what will happen to the cell membrane? The membrane will not be intact. It will not be tight. It will become fluid. It can start allowing any material to pass inside and outside of the cell. So that is the physiological importance of cholesterol, a very important class of lipid. And in humans, the cholesterol is synthesized in the liver. That is the structure of cholesterol, which is responsible for giving rigidity to the structure of the lipid bilayer membrane. Cholesterol is insoluble in the blood, so when it is released in the bloodstream, it immediately forms complexes with lipoproteins. Now, cholesterol can bind with two types of lipoprotein. One is called HDL, high density lipoprotein. Another type is called low density lipoprotein. Supposed to learn about HDL and LDL, we need to know the function of cholesterol. To know the function of cholesterol, we need to know what is a lipoprotein. A lipoprotein is a spherical molecule with water-soluble proteins on the exterior. Therefore, when cholesterol is bound to a lipoprotein, it becomes blood-soluble and can be transported throughout the body. What does HDL also do? HDL cholesterol is transported back to the liver. If HDL levels are low, then the blood level of cholesterol will increase. That is why HDL is given the name of good cholesterol, because it will bring back the cholesterol to the liver and prevents the increased levels of cholesterol in the blood. High blood cholesterol level is associated with plaque formation in the arteries, which can lead to heart diseases and stroke. So cholesterol intake from the diet enters into the bloodstream in the LDL form. This helps why consumption of foods with high cholesterol content can lead to increased blood levels of cholesterol, which is bad for health. So reducing the cholesterol in the diet can lower the blood level of cholesterol. This can reduce the amount of plaque formation. Is there any other methodology? The statins, which are prescribed for cardiovascular diseases, prevents the cholesterol formation. And aerobic exercises can also contribute to health by increasing the HDL levels in the blood, which can help in the transport of cholesterol from the blood to the liver. To understand the function of lipids, we need to know a little more about the digestion and absorption of lipids, because ultimately we can understand the function only if we know how the lipids are digested from the food we take and how they are absorbed and how they reach their destinations, how the body prepares the specific types of lipids required for its own different physiological functions. So we need to first understand the digestion and absorption of lipids. The bile salts, which are formed in the liver and secreted in the bile, they enable the emulsification of the products of lipid digestion. So it is like this. The lipid digestion starts with the action of the enzyme lipases, which break down the fatty acid esters, and cholesterol ring is also broken down by other internal milieu. Now the bile salts, which are formed in the liver and secreted in the bile, go and emulsify the products of the lipid digestion. Now what happens? They all aggregate and form micelles and liposomes. They form micelles and liposomes of both the phospholipid component of lipid and the cholesterol from the bile. Because the micelles are soluble, now we have converted the insoluble lipid into a soluble micelle. They allow the products of digestion, including the fat soluble vitamins, to be transported to the aqueous environment of the intestinal lumen, and they permit close contact with the brush border of the mucosal cell and helps in the absorption. The bile salts pass on to the ileum, where most are absorbed into the enterohepatic circulation. In starvation, esterified lipids from the circulation or in the tissues are oxidized, as well as particularly in the heart and skeletal muscle cells, where considerable stores of lipids are to be formed. So now we have with ingested lipid in the food, broken down by lipase enzymes and other internal milieu, pH and other things, then they form micelles with the bile salts secreted by the bile, secreted by the liver and secreted by the bile. They all form micelle and a liposome. So the free fatty acid uptake by the tissue is related directly to the plasma free fatty acid concentration, which in turn is determined by the rate of lipolysis in the adipose tissue. So after dissociation of the fatty acid albumin complex at the plasma membrane, the fatty acid binds to a membrane fatty acid transport protein that acts as a transmembrane co-transporter with sodium. Now cholesterol and phospholipid we saw. Now we need to understand how the triglyceride is transported. Now I am going to introduce to you a new terminology called chylomicron. The triacyl glycerol, we know how phospholipid and cholesterol are transported through micelles and liposomes. Triglycerides are transported from the intestines in chylomicrons and from the liver. Very important lipoprotein called very low density lipoprotein. Phospholipid and cholesterol, high density lipoprotein and low density lipoprotein. For triglyceride, very low density lipoprotein. So what are chylomicrons? They are found in chyle, which is formed only by the lymphatic system, and they are responsible for the transport of all the dietary lipids into the circulation. Small quantities of VLDL are also found to be in chyle. However, most of the VLDL is of hepatic origin. That means the synthesis is from the hepatic. They are the vehicles for the transport of triacyl glycerol from the liver to the extrahepatic tissues. The lipoproteins are very, very essential for absorption and digestion of lipids. Let us see quickly the classification and functions of lipoprotein. Cholesterol and other lipids are carried on plasma lipoproteins. Apolipoprotein, designated protein in its lipid-free form, combine with the lipids to form the several classes. So before combining with the lipid, it is an apoprotein. When it combines with the lipid part, it becomes an apolipoprotein. Our lipids of interest, triacyl glycerol, phospholipid and cholesterol. So if phospholipid, cholesterol and triglycerol have to perform their function, first they have to be properly come into circulation. So we need to understand their digestion and absorption, for which a wide variety of lipoproteins are available. Chylomicron, the source is from intestine. The main lipid component is triacyl glycerol. Chylomicron remanence, the source is the previous chylomicron. The main lipid components are triacyl glycerol, phospholipids and cholesterol. They help in the transport of this. VLDL, the source is liver. They help in the transport of triacyl glycerol. IDL, intermediary density. Where does it come from? It comes from VLDL. It helps in the transport of triacyl glycerol and cholesterol. LDL, low density lipoprotein. The source is VLDL. It helps in transport of cholesterol. HDL, the source is liver, intestine, VLDL and chylomicrons, and it is important for the transport of phospholipids and cholesterol. Now that we have understood the important classification and functions of different lipoproteins, which are responsible for the digestion and absorption of major lipids like triacylglycerols, phospholipids and cholesterol, whose functions we already have clearly understood in several modules and also in this module. Before we conclude the functions of lipids, I would like to introduce you to another important kind of a lipid fatty oxide derivative product called ketone body, the types and its importance. Why should we know about it? The understanding of lipids, their characteristic functions and properties will not complete if we do not understand about ketone bodies. The ketone bodies are formed in liver to free up the acetyl-CoA for beta oxidation, metabolized in other tissues including brain as an energy source. The formation of ketone bodies is a consequence of prolonged metabolism of fat. When does it happen? Let us assume that there is a starvation situation. There is going to be a prolonged metabolism of fat. The fatty acids are going to be oxidized for generating energy, equivalents and reserves. The formation of ketone bodies in the liver actually enables liver to metabolize even more fat, but freeing up CoA that would otherwise be tied up as acetyl-CoA waiting to get into the TCA cycle. While increasing the metabolism of fat through beta oxidation, much of the mitochondrial CoA pool may become tied up as acetyl or acyl-CoA. Upon prolonged fasting and heavy reliance on fat for energy, the liver induces the enzymes required for the formation of ketone bodies, and brain induces enzyme required for their metabolism. Now, the ketone bodies formed in the liver mitochondria by the condensation of 3 acetyl-CoA units. 2 acetyl-CoA units are first condensed to form a acetoacetyl-CoA. The acetoacetyl-CoA is then condensed with another acetyl-CoA to give HMG-CoA, hydroxymethylglutyryl-CoA. This is then split by the HMG-CoA lyase enzyme again to acetyl-CoA and acetoacetate. The hydroxybutyrate which arises from the acetoacetate by reduction, and the overall sum of ketone body formation is the generation of the acetoacetate and the freeing up of the 2 CoAs that were trapped as acetyl-CoA. Why is this ketone body essential? They are utilized in other tissues by converting the acetoacetate to acetoacetyl-CoA and then converting the acetoacetyl-CoA to 2-acetyl-CoA. What will happen? They will get burned in the muscle mitochondria. They can enter into citric acid cycle. They can help in the generation of ATP molecules. So indirectly, ketone bodies also derivatives of lipid metabolism having an important function. Increased fatty acid oxidation is a characteristic of starvation and of diabetes mellitus, leading to an excessive ketone body formation by the liver, which is called as ketosis. So thus, in this module, we have learnt about the functions of triglycerides, the storage form of fatty acid in the body, phospholipids, important component of the lipid bilayer membrane, cholesterol, a very important lipid, again an important component of the lipid bilayer membrane, and which is also essential for the production of sex hormones, mineralocorticoids and glucocorticoids. What else have we? We have not focused our attention on understanding the functions of other type of lipids like sphingolipids, cerebrolipids, cerebrosides, gangliosides. We have learnt about them in detail in module 15, but we focused in this module on the lipoproteins, which are also an essential component of lipid metabolic pathway, which help in the transport of lipid and maintenance of their levels. And finally, we have also understood about ketone bodies, a type of lipid metabolic derivative product that results from lipid metabolism and its usefulness in helping in generation of acetyl-CoA for further energy release. Needless to say, we need to know that all these lipids have very, very important role, physiological and biochemical role to play for normal healthy functioning. And the underlying take-home messages: we need to only focus on the quantity and the levels in which they have to be maintained. But we do essentially need lipids.